When it comes to thinking about redox, there's often too much emphasis on the ox without really thinking about the electrons involved. And well, those electrons are what's really defining redox. So I like the mnemonic oil rig. Oxidation is loss of electrons and reduction is gain of electrons. Now, often, but not always, oxidation is going to correspond to the gain of bonds to oxygen, and the reduction is going to correspond to gain of bonds of hydrogen or loss of bonds to oxygen. And so, yes, it's kind of helpful to think about, okay, oxidation, more bonds to oxygen, reduction, less bonds to oxygen. But thinking of it that way is going to get you into trouble when things aren't quite so simple. And more fundamentally, you're missing the point of redox, which is about those electrons. And especially when we talk about metabolism, when we talk about catabolic pathways, and we think about all these oxidative reactions that are taking place, and these electrons, the electrons are what's getting passed. The electrons are going to get passed to NAD plus to give us NADH, which then we can cash in for ATP in oxidative phosphorylation. And so all of that is going to be taking place because of the transfer of electrons. And so let's step back and think really, what is redox all about? How can we track these electrons? Where are these electrons going? Well, how can we determine whether redox is actually occurring? We can do things like using oxidation numbers, so I'll show you how we can calculate these, as well as show you how this all applies to metabolism. So let's dive in and talk about things. It's a lot easier to use those kind of like simple mnemonics than to think about the electrons, especially when we're thinking about biochemistry, where unlike if you might have learned about redox in like general chemistry, where you're actually seeing whole electrons moving and things like this, in the case of biochemistry, things are a little more hidden. So instead of whole electrons moving, you can think about gain and loss of electron density. And so you want to look at new or change bonds to an atom and see if the electrons are now getting pulled towards or away from it. If you're getting shared electrons pulled away from you, well, now you've been oxidized. And this is often, but not always, going to occur when you form bonds to oxygen because, well, oxygen is really good at pulling those electrons away. When we talk about the ability to pull electrons, we're talking about electronegativity. And oxygen is going to be able to pull those electrons away, and thus the atom is going to have been oxidized. If, however, the shared electrons are getting pulled closer, the atom has been reduced. And well, if you think about when you can pull electrons closer to you, it's well, when if you bind something that's less electronegative to you. And who's the weakest when it comes to electronegativity, who's really, really bad at holding those electrons? The hydrogen. And so if you form a bond to hydrogen, that hydrogen isn't going to be very good at holding onto its electrons, and so you're gaining electron density in that way. Oxidation and reduction go together. If you're pulling electrons from one thing, you're pulling it to one thing. And so the thing that gains electron density or gets reduced had to oxidize something in order to do so. And so we call the first thing the oxidant or the oxidizing agent. The thing that loses electron density, the thing that's getting oxidized, had to reduce that other thing in order to do so. So it's called a reductant or a reducing agent. So how do we determine whether something's a reductant or an oxidant and what the heck is going on? For this, I like to turn to a little dog analogy. So if we think about those atoms as kind of having the protons are going to be held in the central nucleus, and then we have the electrons around it. And I like to think of these electrons as dogs and the valence electrons especially. So the valence electrons you have in your outer shell, and these are the electrons that are kind of going to be the most reactive. These are the ones that are furthest from the pole of the protons in the nucleus. And those protons are kind of like dog walkers that are holding onto them in leashes. The more electronegative the atom is, the kind of tighter those leashes are going to be. This is going to be a combination of how many of those protons they have. So those are the polars, like how much pulling strength they have, as well as how far away those dogs are. So how far out are those valence electrons? And so you have this trend where you go to the upper right corner of the periodic table, and that's where you're going to find the most electronegative things. And so we see that over there, we've got our oxygen, we've got our nitrogen. Sulfur is not too bad. So all these are going to be more electronegative, especially more electronegative than something like our hydrogen. And so what's going to happen is that those electronegative atoms are going to pull more on those those dogs. And so when you're sharing those dogs in a covalent bond, because that's what a covalent bond is, it's dog sharing. Okay, well, what it really is it's the sharing of the electrons. But if we're saying those electrons are dogs, so you can think of two dog walkers walking side by side. One of the dogs starts sniffing the other dog's butt. And now you know it, you have these two walking side by side. And well, who does the dog like more? Does it like the first owner or the second owner? And therefore, you can kind of determine, do we get a shift in our loyalty of the dogs, in which case we would have one of the molecules being reduced and one being oxidized, or do we not have any change at all? If you have a fully covalent bond, well, here you're going to have fair sharing between them. You can think of both of the protons being kind of like equally good at wooing the dogs. And so those electrons are shared fairly. If you were to split them up, each owner would still have a dog. But now imagine that you have a situation that is not so fair. Like maybe you have that oxygen, which we said was really electronegative, and it's bonded to a hydrogen, which we said was like really, really not electronegative, electropositive. And so what's going to happen is that the dogs are going to, if you were to split these up, the dogs are going to stay with that oxygen. They've changed their loyalty. And therefore, if we were to compare the state of this hydrogen and the state of the oxygen before and after, well, if we think about in a fully covalent bond, if we have this fair sharing, well, here each of these hydrogens owners was going to have an electron, and yet here it doesn't. And so what would have happened was that, well, going from that pure hydrogen to the water, the hydrogen is going to have lost electrons. And when we say losing electrons, we say it's oxidation. But the oxygen, well, if you compare just like pure oxygen, O2, you would have fair sharing between those because that would going to be one of those fully covalent bonds. However, now you get two electrons, you've got one from each of those hydrogens. Voila, you've gained electrons, you've been reduced. Do not confuse hydrogen with a proton. Protonation and deprotonation are not redox. By definition, a proton has no electrons. Redox, remember, is all about the electrons. If we don't have any electrons, we're not doing redox. So remember, we said that if we split these dogs up, we would still have the electrons stay with the oxygen. So it's like the oxygen already won those electrons. And so now if we're saying that the hydrogen is just leaving, that's what we said was theoretically happening before, was hypothetically happening before. And now we're saying, okay, let's actually go and actually do it. That's not going to change things. You already had that reduction happen and that oxidation happen before you lost that proton. Losing the proton is not changing anything in terms of your oxidation or your reduction. So protonation and deprotonation are not redox reactions. So in the case of deprotonation, we have the dog walker leaving without the dog. In oxidation, a dog and not just the walker leaves, or the walker gets swapped with a dog that's harder to woo. So for example, if you went from having that oxygen bound to hydrogen, which it could kind of take nicely, to having a bond to another oxygen where now it has to share fairly, that would be an oxidation. And similarly, if we went from having this pure oxygen where it was sharing fairly, and then when it split, it would only get one from the bond, to having a bond to a hydrogen, well, now it would be basically gaining the electrons from the hydrogen. It would be being reduced. And we think about those were kind of like hypothetical situations. But in the case of an ionic bond, well, here what's actually happening is you are getting that whole that split actually changing. So we could think about the hypothetical changes as being what would happen if those bonds were ionic. And this gets us to a concept called the oxidation number. The oxidation number says, if those bonds were ionic, so if we were to split those atoms up, what would the charge on each of those atoms be? In order to figure this out, you need to take into account how many electrons the atom has in its neutral form, so how many valence electrons it has in its neutral form, and then subtract how many it would own if the bond were ionic, and that'll give you the oxidation number. And to figure out how many it would own, you need to take into account which of the atoms is going to be more electronegative and thus win the electrons. And so you have where you have the oxygen is the most, then nitrogen, sulfur, carbon, and hydrogen. For the purpose of this class, I'm not going to actually make you calculate the oxidation number, but I do want you to understand some important things about it. One is that the higher the oxidation number, the more oxidized something is, and the lower the oxidation number, the less oxidized it is. And if we think about why this is, well, electrons are negative, and so if you have more electrons, that's going to make your oxidation number lower. And if you have a lower oxidation number, that's actually going to make you a better fuel because it means you have more electrons you can give up. So if we think about some fuels, if we think about like methane, we have carbon in its fully reduced form, it's going to have a -4 in its oxidation number as opposed to something like carbon dioxide where we've got it fully oxidized. Now we have a +4. We've given up all of those electrons. And so now if we were to split these up, all those electrons would go to the oxygen. Our carbon has basically lost them. This would not make it a good fuel, whereas something like methane would be a good fuel. Similarly, we're going to have things happen in biochemistry where we're going to be oxidizing molecules in order to gain energy by transferring the electrons from those oxidations onto biological carriers such as NAD plus and FAD. Then we can cache these in for ATP. So if you see an increase in the oxidation number, something's being oxidized. If you see a decrease, something's being reduced. And if you're seeing a protonation or deprotonation, that's not going to change your oxidation number because that is not redox. But if you see a change in those oxidation numbers, that is going to tell you that some sort of redox is occurring. A couple other just quick things about oxidation numbers is that the sum of the oxidation numbers should always equal the formal charge, and that in its atomic form, an oxidation number, or its like molecular form, an oxidation number is going to be 0, because, well, if you're sharing fairly with something of your same type, and then you split up, you're going to be stuck with the electrons that you started with, which would be the number of valence electrons, and so that's not going to change. Also, if you want to learn more about oxidation numbers, I have a separate little video and I have a longer version of this video, which goes more into depth into actually putting these oxidation numbers into practice. I find them helpful, but I don't want to make you do too much. So I'm not going to make you do oxidation numbers, but that content is there if you are interested. You might also realize that we're typically moving by numbers of multiples of 2. So we're going from -4 to -2 to 0 to +2 to +4 when we think about the carbon's oxidation state. And one of the reasons why this is that we are basically losing 2 electrons in each of these changes. Let's think about going from methanol to formaldehyde. We had to remove this proton. So we lost a proton, and then those electrons could kind of shift down here to form this double bond. But we would have we would have too many bonds to this carbon. And so we have to actually remove this hydrogen with its electrons. If we think about a hydrogen with its electrons, that's what we call a hydride. So we need to lose this hydride and a proton to go through each of these. And because we're losing 2 electrons, well, now our oxidation number is going to increase by 2. Where are these electrons going? These electrons are often in biochemistry, these electrons are going to be going to NAD+. And this is going to leave you with NADH. So we get this transfer of the hydride to the NAD+. The NAD+, the plus refers to the charge on the nitrogen, not to the charge on the NAD+. If we think about NAD+, well, overall, it's still going to be negatively charged, but this nitrogen is positively charged. And the reason why we emphasize this is this makes the nitrogen really want electrons. And so it's going to serve as a sort of electron sink. In this ring of the NAD+, what you have happen is the hydride is going to add on to this. This pushes the electrons around the ring onto the nitrogen, and this makes the nitrogen happy because you're giving a positive thing negativity. That's going to neutralize that, make that all happy. Voila, you've got your hydride transfer. And then remember, too, that we had that proton that we had to lose. You can think about things a couple different ways. If you think about the electrons from this oxygen flowing down to form a double bond, now what's happened, and you kick off the hydride, you would have a positive charge here in which your base could remove, because that would not be very happy. You can also think about the base removing the hydrogen beforehand, then the electrons flow down, and voila. In either case, once the base is protonated, well, either the base could be water, or it could basically then lose the proton to water. free solution. Anyway, that's how you get that kind of plus, that proton. If you think about going from a carbonyl to a carboxylic acid, well, here we can think about, okay, well, if this hydride were to leave, we would have a positively charged carbon. That wouldn't be happy. So a water could attack it, and then you would get this OH and a proton. Voila. Let's think about how this happens in our malate dehydrogenase. So remember malate dehydrogenase, it takes malate and it takes electrons from malate, puts them onto NAD+, leaves you with oxaloacetate and NADH. And of course, it can go the other way as well. How does it actually do this? Well, we can see that what's going to happen is you have the cysteine in the active site with this nitrogen that is just poised and ready to take a proton from this malate from the alcohol group of the malate. When it does this, it allows for the hydride to be transferred to the NAD+. The electrons are going to flow around the ring to the nitrogen. That plus charged nitrogen is now not going to be plus charged. It's going to be happier. And voila, you're left with NADH, as well as with, now you have oxaloacetate. But you have the cysteine be protonated, which wouldn't allow the reaction to go anymore. So you have to have a water or something come and deprotonate that. And then that's how you get that proton released. It's not just in this malate dehydrogenase reaction where you have pass-offs of electrons to NAD+. You see it all over the place in metabolism, including in glycolysis. So glycolysis uses up NAD+, and it generates NADH. In order to keep glycolysis going, you're going to have NAD+ available. Not only do you need it for the reaction to happen, but NADH is going to inhibit some of the enzymes that are needed for catabolism. And so your body has ways in which to regenerate that NAD+. Either you can use anaerobic fermentation to make like lactic acid if it doesn't have the oxygen, but if it has oxygen, it can go through, and it has the electron transport chain available to it. It can go through that, and then it can regenerate the NAD+ that way. So you need that high NAD+ to NADH ratio for glycolysis to happen. NAD+ is good because having a bunch of it is going to make your reaction more thermodynamically favorable. If we think about thermodynamics and free energy, remember the favorability is going to depend on the ratio of products to reactants, because we want our products to reactants ratio to be low so that we have more reactants and are driving towards products. If we say that our products here would be NADH and our reactant would be NAD+, we would want this number to be very small. So we would want to have a little bit of products and a lot of reactants, which would mean having a low ratio of NADH to NAD+, and that'll help make our reaction more favorable. However, what's not so good is then if you want to actually go and reduce things. And so if you want to reduce things, you want to have a different stockpile. And so this different stockpile that we often use is NADPH. It's got that same ring and all that great stuff that NADH had and that NAD had, but we can kind of flip the ratio around and keep a lot of the NADPH form, the reduced form, as opposed to the oxidized form. And if we have a bunch of the reduced form, now we can use it to reduce other things. One of the other things that we use it to reduce is a molecule called glutathione, which is a cellular-like antioxidant. And so now let's talk a little bit more about glutathione and when our bodies use it. This goes back to one of the reasons why I think it's important to think about the electrons and and actually calculate oxidation numbers and stuff, rather than just focusing on the simple rule that bound to an oxygen is typically going to be an oxidation reaction. But of course, the first thing I'm going to show you is that a bond to an oxygen is going to be an oxidation reaction. In proteins, one of the oxidation reactions that often occurs is going to be the oxidation of cysteine. Why does this occur? Well, because that sulfur can get oxidized, especially if there's like reactive oxygen species around. That was bound to oxygen, but it turns out that sulfur is also electronegative, and it's more electronegative than carbon. It's more electronegative than hydrogen. And so it's not as electronegative as oxygen, but if you swap out a bond to a hydrogen to a bond to another cysteine, well, now what's going to be happening is that is going to be oxidation. You go from getting those electrons from the hydrogen to having to share fairly with the other sulfur. And so what's going to happen is now you're going to have had an oxidative reaction occur. So cysteines can form these sort of like crosslinks within a protein or between proteins, and this can be controlled for making protein, like sturdifying proteins, or it can be uncontrolled when you have like reactive oxygen species happening. When you form these crosslinks, so these disulfide bridges, they're sometimes called, that is going to actually change what we call the cysteine. So we go from having like cysteine to having cysteine. And so basically you lose the electrons and you lose the E in the name is how I remember it. And so that would be oxidation when you are going and forming these crosslinks because you're taking that bond to hydrogen where you got to take the electrons to a bond to a sulfur where now you have to share electrons. So that's going to be an oxidation reaction. Kind of like we saw when we had our OH versus we had the O2. When you're sharing fairly, you're getting basically losing electrons compared to if you got electrons from the hydrogen. However, we can also think about with cysteine, if we were to go from a thiol, so the protonated state, to the thiolate, the deprotonated state, this is not going to be a redox reaction because, as we said before, that hydrogen was already basically giving up its electrons to the sulfur in the hypothetical scenario. And so now we say if that actually happens, then the sulfur is not going to be changing its oxidation. How do we undo these crosslinks? In our bodies, we use a molecule called glutathione. And glutathione is kind of made up of amino acids, and it serves as a source of reducting power. It's like our antioxidant that our cell uses the most. It's able to reduce these crosslinks by forming sort of like mixed disulfides in the process. So this is actually showing you how this happens with DTT. So DTT is a commonly used agent in the lab to reduce crosslinks. We want to reduce these crosslinks because they do things like mess up our protein gels. If you get those like weird reactive oxygen species modifying the protein or causing crosslinks between different parts of the protein that you don't want. Also, some multimers are held together by disulfide bonds. Not all of them, but some of them. And those ones that are held together by disulfide bonds wouldn't be able to be separated with just heat and SDS. So instead, you have to also add the reducing reagent. And so that's why we added the beta-mercaptoethanol to the loading buffer for our SDS page gel. Speaking of having disulfide bonds in proteins, typically you're going to find them in proteins that are secreted because it's going to allow the proteins to be like sturdier. But because we try to prevent unwanted crosslinks inside of cells, it's going to be a reducing environment inside of our cells, and so that you wouldn't have just like nilly-willy crosslinks forming. And the crosslinks you do want to form, you actually go through special processing pathways inside of the cells to kind of protect them from that reducing environment until they can get where they need to be. And so glutathione, really great. But glutathione, in order to reduce those crosslinks, it becomes oxidized in the process. And when the glutathione is oxidized, it's no longer useful for reducing things. If you want to use the glutathione again, you need to reduce it. So how are we going to reduce it? Well, remember we said in our bodies, we typically use NAD plus to oxidize things, but we wouldn't want to use NADH to reduce things because we purposely keep a really low concentration of NADH. Instead, we want to turn to something different. We want to turn to our NADPH. Our NADPH is really good because we can keep a high concentration of it. We can regulate it separately from the NAD+. It still has that same reactive part that's good for these redox reactions. But here you've got a difference. You've got this extra phosphate group, which means that enzymes can kind of like tell it apart, and you can regulate them separately. We can keep this high proportion of NADPH as opposed to NAD+. And now we have this stock of antioxidant in our power to reduce this, that glutathione to regenerate the antioxidant NADPH. And then we can use this to combat those changes that happen to those cysteines and things because of reactive oxygen species. Or we can kind of take the hit before those other things, before things get too bad. And so voila, NADPH, great. NADPH is also really useful for anabolism. So if we said that catabolism was the breakdown of molecules and it was often oxidative, Well, anabolism is the reverse. It's the making of molecules, and it's often going to be reductive. And so we see NADPH be used in the synthesis of lipids. So our bodies use NADPH and glutathione to help reduce things. At the salon, you might use a molecule called ammonium thioglycolate. Cysteine crosslinks hold together proteins in your strands of hair. So the protein keratin is really abundant in your hair, and it's got a lot of cysteines. Those cysteines form crosslinks, and depending on how they form those crosslinks, you might get really curly hair. If you use this reducing agent, you break up those crosslinks, and then you can add an oxidizing agent like hydrogen peroxide. Once you've kind of put the hair in the shape that you want it in, that's going to allow these new bonds to form that hold in place this new shape, as opposed to the old shape that you didn't want. So if you were to break those bonds, then curl your hair up around curlers, then add this oxidizing agent, well, now those crosslinks are going to form, but they're going to form in the form that's going to hold together that new style. All of that has to do with redox. Oxidation, loss of electrons or electron density. Reduction, gain of electrons or electron density. And remember that we could formalize that with oxidation numbers if we wanted to. A lower oxidation number is going to be more reduced. A higher oxidation number is going to be more oxidized. And this is calculating oxidation numbers can be a helpful strategy to help you realize when redox is and is not occurring. But I won't hold you accountable for doing it. I will hold you accountable, however, for remembering your oil rig. Oxidation is loss of electrons. Reduction is gain of electrons. Protonation and deprotonation do not involve electrons, are not redox. By definition, a proton has no electrons to give or take. But our biological fuels do have electrons to give and take. And so we see these